Welcome to PPLS Perspectives, a podcast bringing students and academics together to explore the research carried out by the University of Edinburgh's School of Philosophy, Psychology and Language Sciences and the impact this work has on society.
Hi everyone, and welcome to episode 4 of the PPLS Perspectives podcast, where we aim to shed a light on members of the School of Philosophy, Psychology and Language Sciences here at the University of Edinburgh via an informal discussion of the importance and the impact of their work. My name is Kate Nave. I'm a postdoctoral research fellow in the philosophy department here at the university, and I work on the differences between living organisms and machines and the limitations of mathematical modelling when it comes to the former. These themes also nicely connect with the work of our guest here today, Mazviita Chirimuuta, who is a senior lecturer in the philosophy department. Prior to coming to Edinburgh, Mazviita was a member of the Department of History and Philosophy of Science at the University of Pittsburgh, but further back in time she also gained a PhD in neuroscience and worked on the mechanisms of colour vision. That experience, as we'll hear, has informed her philosophical work, in which she not only looks at how neuroscientific research can provide insight into philosophical questions, but also interrogates neuroscientific practice through a philosophical lens. Today, we're here specifically to talk about her recently released book, The Brain Abstracted, which deals with the challenges of simplification in neuroscience. So, welcome to the podcast, Mazviita.
Thank you very much. Yeah, it's nice to have this opportunity to discuss my research.
So this podcast has a pretty consistent format of 5 standard questions with some digressions, but we're going to begin with an elevator pitch in which the aim is to explain your work and its key points in the time it takes to share an elevator with someone. Fortunately, we both work in a 14-storey building with a very old elevator, so that's a good chunk of time. But if you could just begin by giving us an introduction to your research and particularly the topic of your book.
Yeah, sure. So I'm a philosopher of neuroscience. So philosophy of neuroscience, it's a subdiscipline within the philosophy of science, where we look at the questions about how knowledge is generated and communicated within the scientific fields. And there are many different branches of biology, neuroscience, etc., in which the different challenges of how scientists have to do their experiments and actually find systems in nature which enable them to explain and make sense of various phenomena and leading to technological innovation. So philosophy sort of takes a step back from that and tries to make sense of the different procedures and methodologies within science. So philosophy of neuroscience is a particularly fascinating instance of this, in particular because of the relationship between the brain and mind and the questions about how scientific research on the brain helps people understand what's going on in their own minds. So there's a related field to philosophy of neuroscience, which has been since the 1980s known as neurophilosophy. And there the idea was actually to take the results of neuroscience and try and figure out how those results could shed light on very old philosophical questions about the nature of the mind, about what it means to be an agent, what it means to have free will, what consciousness is, etc. And so my book project is actually a bit of a response to neurophilosophy, in that it's saying that if we look in detail at the methodology of neuroscience, we see that there's so much that has to be done in terms of simplification, abstracting away from all of the complicated, messy details of what actually goes on in the brain, and presenting both scientists and members of the public with almost completely clean-cut and almost distorted versions of what's actually going on in the brain that are fine for various purposes within science. But if you're a neurophilosopher and you want to just ask questions about, well, what is the nature of cognition or consciousness really, and you just take these neuroscientific results at face value, you get something of a distorted picture. It's somewhat misleading. So that's the argument that I'm presenting overall in my new book.
And you begin the book with this rather wonderful survey of just all of those messy details and how complex the brain is. And there's a lot of fantastically large numbers there, 85 billion electrically excitable cells in the brain alone. But, if we look at a beach, there's many grains of sand, but no one marvels at the complexity of, say, a beach. So could you say a bit about what it is about the brain in particular, the kind of features that you draw out that make a system complex?
Yeah, so one of the really important features there is just the variation from one individual to another, or within one brain from one cell to another. If you look at a beach with millions of grains of sand, any one grain of sand is quite similar to the next one. The minor differences in chemical composition, size and shape, etc., don't make a big difference to what you might want to do with sand, or if you like, the functionality of sand, the properties of sand taken on aggregate. But if you look inside the brain, each individual neuron is kind of as different from the next one as, you know, each tree in a park. You know, how if you look in the park at the different trees, each individual tree has a different shape. It's sort of due to how it's grown, how it's been pruned over time. Neurons are very much like that. There's not a lot of identity between different neurons. They have different structures. There are cell types. You can classify them into cells of this sort versus another sort, but in their fine anatomical structure, they're actually very heterogeneous. And this is related to their functionality, because they connect with cells around them. That's how neurons communicate with one another. And so if you just are interested in how a neural circuit works, you can try and, you know, co-classify all of these different neurons and say, well, they're all basically the same type and they do the same thing, like grains of sand. The differences don't matter. That's what neuroscientists do very much of the time. But there is something actually quite different from the grains of sand going on, because some of those fine structural details do matter, just in the way that, you know, a tree which has grown a certain kind of branching in one direction, it's related to what's going on in its surroundings in terms of light, in terms of nutrients, and that's a similar thing that goes on in the brain. So there's that variation from one, you know, within the brain, from one individual cell to another, also from person to person or animal to animal. There are a lot of differences that mean that you can't just average over all of your results and expect that you're going to have a very representative sample of what's going on if you just take average data all of the time, even though for many scientific purposes you do find that you have to take average data. And then there's also a lot of variability across time. So the brain is always changing. This phenomenon of neuroplasticity, which shows some large scale and some local reorganization and changes in connectivity patterns, changes in responsivity that goes on from time to time due to experience or maybe even due to injury, means that the brain's not a static fixed target. What happens to a person from one day to another kind of changes the physiology of the brain. So that makes it challenging if you're a science where you want repeatable, reliable results. What happens with experimental methodologies is that you want to try and constrain responses in such a way that you maximize the amount of repeatability that you can get, but then what you're kind of moving out of your picture is actually how much plasticity there is and how much that is actually really important to the functionality of the brain.
Mm-hmm. And you mentioned kind of analogies to things like trees as being another system where you see this kind of variability. I was wondering, to what extent do you think this kind of variability and heterogeneity is unique to the brain? Is it something that we see in other systems as well, and is the difference just one of degree, or is there something really quite fundamentally different about the brain alone in that regard?
Yeah, so I don't think it's fundamentally different about the brain. I think this is something that we see pervasively within biology. So yeah, like trees are growing all the time, they're changing their structure all the time, other plants are doing that. You see differences from one individual to another in all kinds of different physiological parameters, anatomical features. I would say brain cells, if you look at their cell types, they're probably more heterogeneous than liver cells, kidney cells, muscle cells, for example, and I think that is related to the specific task that the brain is involved with, which is cognition, ultimately providing the basis for intelligent action. So if you think about what intelligence allows people and other animals to do, it allows them to respond sensitively and in a coherent way to the challenges of a changing environment. So the brain itself as an organ has to be extremely context sensitive. It has to be sensitive to all of the different dynamics and fluctuations in the world around us in order to allow for adaptive behaviour to those challenges. So that means that these features of plasticity and heterogeneity are maybe more pronounced in the brain compared to other organs of the body, but I see them as, if you like, an exaggeration of plasticity and variability that we see pervasively within biology. In contrast, if you look at the systems studied in the physical sciences, they do tend to be much more homogeneous and much more stable in their properties. So if you think about electrons, the characteristic responses of electrons don't change whether you're in one country to another, whereas the characteristic physiology of pretty much any organism will be somewhat dependent on the environment that it's grown up in. Plants, their characteristics of how they grow dependent on the environment around them. Ecological systems in general change from one year to another, dependent on a whole range of context-dependent factors, which, if you're a physical scientist, there's just nothing as comparable to that. Physical non-living objects are just not as sensitive to their environment as living things inherently are.
And when you speak about the challenges of trying to get repeatable experimental results with a system like that, I think one of the things that is very interesting about this book is that you're not just talking about that from a theoretical perspective. You've had the experience of being someone who is trying to get those nice, clean experimental results. And you talk about that a bit in your book. When you were working on the early visual system. I was wondering if you could tell us a bit about what that's like as a neuroscientist, being faced with that kind of unruly system.
So I started out my research career actually in neuroscience, a branch of neuroscience which is studying the visual system specifically, with some overlap with psychology because we were doing behavioural experiments on people. I was my own experimental subject for a number of years. So I only moved into research in philosophy after I finished my PhD and did some postdocs in that area. So the task that I had during my PhD was studying the sensitivity of the human visual system to fine gradations in contrast. And for those experiments, our core stimuli were these very simple black and white stripy patterns known as sinusoidal gratings. And there's a theory that was predominant at the time, which is trying to analyse the responses of the early visual system, primary visual cortex, to these sinusoidal gratings of different thicknesses, widths of the bar, and different orientations. And the theory was that individual cells in primary visual cortex are sensitive to just one particular set of stimuli of this sort, so a particular thickness of the bars, a particular angle. And the thing is, if you stimulate the brain with very simple stimuli of this sort, what you can do is, in effect, stimulate just a subset of the neurons that are there in primary visual cortex. And you will get more dependable responses in the behavioural experiments that you're doing with these simplified stimuli, because what you're effectively doing is turning off a lot of the other neurons in primary visual cortex that aren't selective to that one particular very simple stimulus that you've chosen. But this is a really artificial way of looking at things. If you look around a normal room, or even in a really plain room that we're sitting in now, you'll find that there are all kinds of features which will effectively stimulate neurons across a whole range of different brain areas, which means that all of those neurons will be cross-talking with one another. And so activity of one group of neurons over here will modulate a group of neurons over there. And this is what generates the lack of repeatability, the context sensitivity and changeability of neurons that you see in those uncontrolled viewing conditions. But by carefully selecting stimuli and viewing conditions to strip away from all of that complexity which is there in the actual environment when we're looking around us normally, then you can generate more repeatable responses. But then you have to ask, is that representative of how the brain operates in the ordinary environment?
Mm-hmm. And since you've moved into philosophy, you've worked on other topics. You've written a book called Outside Colour, for example, which was specifically on the neuroscience and philosophy of colour vision. What made you kind of decide to now return to this problem of simplification at this point? Was there some specific reason to write this book now?
I don't know, to write this book now. It's something that I've been sort of fascinated for a long time, since I began that early stage of my career as a scientist, because it always struck me, the side of our project, which wasn't the experimental side but the modelling side, we were developing these mathematical models of how primary visual cortex responds to our stimuli. And it always struck me that the maths of these models was like just extremely simple compared to what we knew physiologically about how these neurons responded. So it was just, I find it surprising that just a equation with a few parameters could be thought to capture essentially what these neurons were doing. And I thought, well, that's a really nice result if the maths can work out in such an elegant way. But then there was challenges to the transferability of our models into the responses that you get from more complex visual scenarios, as I was just describing. And so this tension between the scientific drive towards the most elegant, simple, straightforward characterizations of a system that you can get, and actually the challenge of making sense of complexity in the real world, that was a problem that has stayed with me for a long time. And more recently then I've been thinking about computational models in neuroscience as a philosopher of neuroscience, so looking at this recent shift towards actually vastly more complex models based on machine learning, and actually seeing how maybe that drive towards simplicity had been missing out on certain patterns of response. I think that was the main push to making this book, focusing on simplification. Sort of, I feel like this is the right moment for it now.
Mm-hmm. And when you mentioned the sort of drive to using these machine learning methods that may be bringing out details that these kind of simplified models wouldn't, are there issues that you see with those on the flip side? They might be more detailed, but are they making other mistakes, or are they raising other issues in a different way?
Yeah, I mean, certainly using machine learning methods, the models are much more opaque and hard to interpret, especially using deep learning artificial neural networks as models of areas like primary visual cortex. You can get models that are predictively more powerful across a greater range of stimulus conditions, but then it's not as obvious to the scientists what the theoretical significance of these models are. And also, as I argue later on in the book, there are also limitations to how neurorealistic artificial neural networks are. There's still a huge gap between what's going on in real brains and what's going on in these simulated versions of neural processing, even though they're often sort of presented to the public as being extremely brain-like.
Mm-hmm. And you contrasted your book earlier, or the approach you take in this book, with this kind of neurophilosophy movement. And I think the book is sort of part of a more general movement of philosophers engaging with the sciences, which is also in some ways a relatively new thing, but in a much more critical way about the actual practice of science. Can you say a bit about how you approach that in your research and what the kind of challenges involved with that are?
Yeah, so I'd say the actual process of researching sort of varied a bit depending on my own experience with the subject. So writing about visual neuroscience, I did draw a lot from my own experience as a researcher, and then sort of following paths of research that I'd seen sort of subsequently in the field that I worked on, but then these transformations that were happening because of deep learning and new modelling methods. But yeah, I would say the chapter in the book about visual neuroscience is sort of really taking my own experience as a researcher as sort of the starting point to how I address that topic. Whereas a few years ago, I became interested in research on motor cortex, and that came about because I was in a different university at Pittsburgh, where there was a lot of research going on using brain-computer interfaces. So people were recording directly from the motor cortex, the part of the brain that goes across the top of your head, which sends signals down to your spinal cord and then your muscles to move your body around. So people were recording from that brain area and using those signals to control robotic limbs and cursors on the screen. These were sort of prototype systems in development as a potential rehabilitation cure. So I got interested in that research more at a distance of not me doing that kind of neuroscience myself, but being able to occasionally visit labs and have conversations in journal clubs with people that were engaging in that kind of research and just reading the literature on it, so trying to get a handle on where the field was at as a non-specialist, and seeing if there were sort of parallel instances of the kind of simplifying strategies that I'd noticed in visual neuroscience happening also in motor cortex, and arguing that, yeah, there's similarities and differences there, but it's a topic also worth looking at through that lens of simplification. And then there's another part of my book which is drawing on historical case studies, where I really had to look at the published literature, the scientists whose work I'm discussing. They're all deceased, but they had a larger impact on the field, and in many cases the historical scientific research had been discussed and critiqued by philosophers of the time as well. So looking at what other philosophers in the past had written about some of these issues and limitations of how we interpret the brain, even historically, I found that really illuminating.
Yeah, I really like the line in your author bio for the book where it has all of my favourite philosophers are dead. That cracked me up a bit. But yeah, I think that's one of the things that I found super valuable about your book, was that kind of historical context that it has. And particularly, you draw a lot on, I suppose, biologists and philosophers from particularly around the kind of early 20th century, who to some extent are, I think it's fair to say, have been a bit neglected in contemporary biology, and at least in Anglophone philosophy, many of whom are new to me. And I was wondering what drew you to that particular point in time, and what you think made it so fertile for this kind of anti-reductionist thinking.
I did find it a really sort of rich source of reflections on how simplification in science works, and also the limitations of what we can understand about living systems if we rely too heavily on these simplifying methods. It is a really rich time historically for biologists themselves really engaging with philosophy of biology, effectively doing the kind of work that philosophers of biology do today, thinking deeply about the scientific methods and how they adapt themselves to different objects of investigation. So in answer to that question of why this particular moment, say from the 1920s to the 1950s, I think there's always many factors with this. I mean, for one thing, the training that scientists got was less specialised at the time, so it was possible for a scientist not only, a professional research scientist, not only to know their own subfield of biology, but to know what's going on across the different sciences, and even to be well trained in philosophy. And one of the things that was happening across the sciences at that moment, early 20th to mid 20th century, was the quantum revolution. So what happened with that is that people realised that the methods that they'd inherited effectively from classical physics, where you think of the world as comprising a set of discrete particles, basically atoms, which are self-contained objects which have inherent properties which are like inherently independent of what's going on in the world around them. A kind of billiard ball universe is the one that's inherited from classical physics, where you have these isolated entities which kind of bounce against one another, and you explain everything sort of bottom up from there. With the quantum revolution, people were saying, well, for physics, that's not right. Reducing everything to these atomistic interactions is just not how the physical world should ultimately be characterized. And then people were also aware of how that notion of this atomistic billiard ball universe had made its way into biology, and so realizing, well, if that's not right for physics, then it shouldn't be right for biology either. So there was this moment in the 1920s, sort of coincident with the quantum revolution, where people were very interested in biology, about the notion of form, structures, and in psychology it was the era of Gestalt psychology. So this idea that entire holistic network of things is what determines the outcomes in both psychology and then in biology, rather than looking at things in terms of their isolated components and then trying to build up from there, which is the reductionist method, which is what I was saying is really a legacy of classical physics.
And given there was that moment, why do you think it's not had more influence on contemporary science? Why do you think that those findings have sort of been ignored?
Yeah, again, that's an interesting question. It feels like a kind of lost moment, because if you look at biology post World War II, it feels like it's really within the worldview of quite a classical reductionist worldview. And I would say a couple of things to that as well. I mean, one of the issues that I emphasise in the book is that for certain purposes, relying heavily on simplifying strategies is the most effective way to get the results that you want. So I emphasise that if your goals are purely technological ones of taking a biological system where you have a high degree of control of its context and you want to generate certain outcomes in a repeatable way, employing these simplifying methods is the most effective way to do that, so long as you have a high degree of control of the context and you can just try and strip away from all of that context sensitivity and variability that would normally be there with a living thing, and just, if you like, manipulate it into a certain regime of behaviour or physiology that you might happen as a researcher to want to keep reproducing. And that has been the dominant method in biotech, in that very sort of technology-driven approach to biology, which I think for various reasons, economic reasons predominantly, has been the focus. Also, post-World War II, you had the molecular revolution in biology, discovery of DNA, and that was, a big win for reductionism to say, okay, well, we have the molecules of life, let's see how far we can go with that, let's see how far we can go with manipulating and explaining living systems just in terms of this molecular level, or going back to DNA and studying individual proteins and cells. And I would say maybe at this time people have pushed that approach as far as it will go. There's still lots of biomedical challenges that have not been resolved using that approach. Plenty of diseases and conditions which are not curable just from taking the molecular genetic perspective. So I think, as always in biology, you see a back and forth between more reductionist and more high-level anti-reductionist approaches, and they have their strengths and weaknesses. Often, if you're wanting to look at things in a very mathematically precise, quantified way, then trying to be too high level, trying to take in too much context dependency, you just don't get the rigour that you get with that more reductionist approach.
And I guess we're coming to our final question, which is just, what kind of impact do you hope that the book will have, not just within neuroscience and philosophy, but also, I suppose, for people outside of those fields when they interpret the findings and interpret what neuroscientists are telling them?
Yeah, I would say actually the real import of the book that I hope to have is not within neuroscience itself. I don't see my task as that to tell neuroscientists how to do their job differently. I mean, given, as I just said, simplifying strategies for certain purposes, they're the logical ones to take. We can't expect them to work all the time, but depending on your purposes, you can justify using them. So I guess amongst neuroscientists, I would encourage them to maybe explore a plurality of results, but it's not my business to tell them exactly how they should run their experiments or what kind of models they should build. So really the message is more for philosophers, people trying to do neurophilosophy and saying these words of caution about how they interpret the results of the neuroscience. They need to be more aware of how much simplification goes into the models and the other outputs that come from neuroscience. And I suppose in terms of the wider conversation, the guiding message is, I suppose, anti-scientistic. So by scientistic, I mean that way of thinking about different situations, different questions and problems, which says that the paradigmatic scientific approaches to those questions are the ones that are best and will exclusively give us the right answers. And I'm saying that if you look deeply into scientific methodologies, you'll see that in some cases, if you're dealing with a very complex interactive system of which human beings and their social groups are, the more you try and use a certain kind of narrow scientific methodology, the more you will leave out of your picture of what is actually there. So if we take a highly scientific approach to the human mind and human society and say, well, neuroscience is going to give us all the answers there, you're actually going to be led into some problems. And so by rejecting scientism, it's certainly not rejecting science. These methods certainly have their place, but it's saying we should be open to a range of methods if we're looking at the human mind in complex real-world situations.
Mm-hmm. Yeah, it's really nice to think of neuroscience as being this kind of focal point for bringing out all of those issues with how we think about our relationship to science in general, I guess. I think that's... this has been the PPLS Perspectives podcast. I've been Kate Nave with Mazviita Chirimuuta. I'd recommend checking out her book. It's called The Brain Abstracted, and it's out now from MIT Press. Thanks for listening.
Thank you very much, Kate.
Thank you.